Hello, this is Marina Magrey again, reporting for Room Now at ACR Convergence 2024 in Washington, D.C. I'm going to report this very interesting abstract which is talking about precision immunotherapy in ankylosing spondylitis. This abstract was presented this afternoon. As we all know, ankylosing spondylitis is a T-cell mediated disease. However, trying to deplete T-cells is not possible because that would put the patients at a very high risk of infections. A subset of T-cells have been implicated in pathogenesis of ankylosing spondylitis. This subset carries this unique receptor T receptor BV9, and these cells are thought to bind with the antigen presented by HLA-B27 positive cells and has been implicated in pathogenesis. In this abstract, a bispecific antibody was developed against this subset of T cells carrying this receptor, TRBV9 receptor. They neutralized these cells, showing to be with using this antibody. The risk of infections was low, cytokine release was low with this binding, so the concern for cytokine release syndrome was minimum, and the recommendation was that by binding and or depleting this subset of T cells, one could reset these autoreactive T cells and produce long-term remission in axial spondyloarthritis and AES. So it was very exciting, and we have to wait till actual human studies are done with this bispecific antibody. There has been a case report from Russia last year which was published showing that these antibodies are work. So something very exciting for the future of axial spondyloarthritis. There was another abstract keeping up with the same theme presented today, which took patients with which had uveitis, acute anterior uveitis, HLA-B27 positive. So they took their ocular cells and blood cells, and they found that blocking this T receptor BV9 T cells may not be enough. They also found another receptor that may be implicated in patients with uveitis, TRBV5. So they were the gist from that abstract was that blocking one receptor may not be enough, and maybe need to block both receptors. Well, this is very early data. Ultimately, needs to be vetted in real human studies and see what works for our patients. Thank you so much.